name is Buse, and my surname is Çevatemre Yıldırım, and I have graduated from biology, actually. And during my childhood, I was always collecting insects and carrying them to my family and showing them and listening the sounds of the insects. So I believe that at that time, I was supposed to be a biologist. And I did my higher education in molecular biology field. And I think the most important part of my career was my postdoctoral research. I moved to another university, Koç University in Istanbul. And there was an incredible team of brilliant scientists. My PhD thesis was on lung cancer, as you mentioned.
Yeah.
And at that thesis, we also focused on drug resistance of the lung cancer cells. And in that postdoctoral research time, my focus was on prostate cancer.
Yeah.
I was also working on drug resistance because tumors sometimes can resist to therapies. And after that, we would ask questions like, how can we overcome this and what are the mechanisms? So the research part actually was here. We are asking questions like, why do these cancer cells resist and what are the mechanisms and how can we target these to make them respond again?
Yeah.
So mainly that was the focus of my last 7 years. So there are several treatment options when it comes to cancer treatment. Maybe you have known, mainly they are surgery, chemotherapy, and radiotherapy. So the most of the patients respond very well to these treatments, but some of them are initially resisting to these drugs, or over time they can stop responding at any time. Yes, the medicines are just stopping to work in those patients. And this is the most challenging part, I believe, in the treatment of cancer. First thing, what we did was mimicking the patient. So we obtained prostate cancer cell lines and make them resistant to chemotherapeutics. And after that, we wanted to reveal their vulnerabilities. In order to do that, we performed epigenetic drug screens because we wanted to know what happens in those cells and what is the mechanism epigenetically that we can target them. Establishing these cancer cells took around a year. And after that, we hit them with a library consisting of 150 drugs. And 3 days later, we just checked their viability, if they are dead or not. So after that, some drugs pop up. So they also clustered. So one was that the protein group that reading the epigenetic marks on the chromatin. So we wanted to focus on that group because there were a lot of drugs accumulated in those class.